Hello, my lovely anatomists and physiologists. We are ready to talk about oogenesis, which is going to occur in the ovary. This is the production of the ovum. And we'll see that it actually occurs inside what is called a follicle. And the follicle is talking about special types of, we can collectively call them follicular cells, and the developing, I'm going to say, gamete. Now, just like we were producing sperm cells through the process of meiosis, we're going to do the same thing in the production of the ovary. So we're going to be, or ovum, I should say. So we're going to be starting out with the oogonia as the stem cell. And my understanding is we do pronounce both O's. And we're going to have the same idea where we're going to do mitosis and produce what's called a primary oocyte, which is also diploid. And then the other daughter cell that we're producing, you're basically regenerating that stem cell so that that is always present. And then what we see happening is that primary oocyte undergoes meiosis 1. Okay, but I'm going to stop right here. What we're going to see happening, I'm going to get a red line, is before we finish meiosis 1, we're stopping. So that this process of going through from an oogonia to a primary oocyte through mitosis, the primary oocyte going into meiosis 1, is happening in the fetal ovary. So that's before this individual is even born. And I'm drawing that red line at anaphase 1. So what this is telling us is that in the infant ovary, at the moment of birth, all of your primary oocytes are frozen in anaphase 1, meiosis 1. Okay? And then what we see happening is we finish with the onset of puberty. So these cells have been essentially kind of like frozen in anaphase 1, you know, from birth until maybe 10 years old, 11 years old, 12, 13, 14, 15, 16. And then what we see happening is when that primary oocyte actually finishes meiosis 1, we're going to call one of those cells a secondary oocyte, and we're going to call the other cell a polar body. And the polar body is basically half of the genetic information. So the secondary oocyte is haploid. But like all of the cytoplasm, all of the organelles and so forth make their way into the secondary oocyte. So it's an uneven divide. And that polar body does nothing. It never is fertilized. It's just totally disintegrated, dissolved by the body. Then we're going to see our secondary oocyte is going to go through meiosis 2. But again, we can take our red marker and we can draw a line. Because meiosis 2 is going to stop at metaphase 2. Okay. It's going to stop at metaphase 2, and it's only completed at fertilization. So when we talk about a cell ovulating, we're ovulating a secondary oocyte that stopped at metaphase 2 of meiosis. So it's not a full functioning gamete that's released with ovulation. And we're going to see we do another one of these uneven divisions when we do complete. Our meiosis 2. We're going to produce another polar body. And we're going to call this fertilized. So I'm drawing my sperm entering. We're going to call it an ovum. And so the ovum is contributing not only half of the nuclear DNA, but all of the cytoplasm, all of the organelles, including the mitochondria, which, if you remember, has its own DNA. So you are inheriting your maternal mitochondrial DNA. And then I'll just say, as soon as you have fertilization occurring, you're going to get your zygote, which is your diploid first starting cell to your next generation. So when the sperm fertilizes the ovum, we get a zygote, which is the start of that next individual. When we go back up here and we see all these stopping points, let's pick up again where we finish our meiosis 2 with the onset of puberty. And let's add in this detail that we'll make sense of later. We're finishing when we get a surge of LH. And that's going to help us actually initiate continuing this particular process. We've said that the polar bodies are going to disintegrate. So they don't have any future. They don't become anything. It's basically a way of removing half of the genetic information. So whereas in the male body and the testes, we're producing 4 sperm for every act of spermatogenesis, in the ovary, we're producing a single gamete for every act of oogenesis. When we talked about that fetal ovary, we said it's going to contain all of these primary oocytes frozen in anaphase 1. So at birth, that infant ovary is going to contain 1 to 2 million potential oocytes. And then at onset of puberty, that number has dropped down to 400,000. And then by the time that individual reaches menopause, there are 0 remaining. 0 left at menopause. This constant reduction of these developing cells is referred to as atresia. As atresia. The other thing that we're seeing happening is in the ovary, not only are we getting this whole process of oogenesis, but we're also going to see follicular development. So we're seeing that the follicles and the oocytes are developing during what we're going to call the ovarian cycle, which is a cycle that's lasting about 28 days. So what we're going to see is not only are you starting out with that infant with all of those primary oocytes frozen in anaphase 1, but you also are starting out with what are described as primordial follicles. So these are, again, present as newborns. And this is basically the primary oocyte, and it's surrounded by cells that are called granulosa cells. So when I said follicular cells, this would be a type of follicular cell. And then we're going to see after puberty, we start to trigger development from a primordial follicle into a primary follicle. From the primordial follicle, we see development into a secondary follicle. And from the secondary follicle, we see a tertiary follicle. And we're seeing that it takes about 2 months for a primordial follicle to go through the development to get to be called the tertiary follicle. And this is basically a fluid-filled follicle surrounding that secondary oocyte. And so my understanding is that during the ovarian cycle, during those 28 days, we're getting, sort of this assembly line where some primary follicles make it to secondary, some secondary make it to tertiary, but we get just one mature follicle. And we can call these Graafian follicles. And this is going to be the one that actually releases the secondary oocyte at ovulation. So we'll actually see a rupturing occurs at ovulation. So the follicle itself will thin out and break, and the ovarian wall will thin out and break. And so you're actually doing a little bit of a rupturing. And I think this is important that we only get one mature follicle, so that ovulation, we're just getting the release of one ovum. This process of ovulation is occurring around day 14 of a 28-day cycle. We're going to go into these details and map it out a little bit. When we talked about atresia, we'll see like 99% of those follicles and those developing gametes are going to experience atresia. So you're going through the process of developing follicles with each monthly cycle, but that also means you're going through a process of degrading a huge number of those with each monthly cycle. When your ovulation is occurring, that means like there's like one winner to the race, and anyone else who was like a tertiary follicle doesn't make it. So those are going to be those follicles that are not making it. When ovulation occurs, this Graafian follicle is going to become a structure called the corpus luteum. And the corpus luteum, we're going to see, is going to release progesterone. Oops. When we talk about that mature follicle and we're getting to ovulation, we're also going to see that that mature follicle is also going to be releasing estrogen. And again, we're going to talk about these hormones as we go through a little bit more detail. All right, so up next, we're going to look at what's happening in the uterus. And from there, we'll start mapping out the hormonal control of both the ovarian cycle and the uterine cycle. So stay tuned. As always, take care of yourselves and each other.